Dallas has over 15 years experience in the industry. Before working at AdAlta, Dallas worked for a company called NextVet, which was acquired. Before that, he was at CSL, so significant experience in manufacturing biologics. Thanks, Dallas.
Terrific, thanks very much. It's not often they let us PD guys out into the community, so I'm looking forward to giving a bit of an outline about product development, manufacturing. So product development is a lead up to coming up with a manufacturing process. I hear some people find this a little bit dry. I've got no idea why. So I'm going to start with the same sentence as I'm going to have at the end for those people who do choose to switch off, which I'm sure is not many, and that is the process is going well, the cell line development is going well, and the purification process is going well. So for all the others, which I'm sure is the majority, I'm now going to enlighten you about manufacturing and why it's so important. The number one thing is regulatory issues. So all the regulatory agencies, the FDA, the TGA, the EMA, they all have codified guidelines and rules which people must adhere to strictly if you want to produce a molecule or a therapeutic which you're going to inject into a human. What's really important about the manufacturing process is the final product, which is going to be a therapeutic, is heavily dependent upon the manufacturing process. If you don't have a robust manufacturing process, then you can't ensure that the product is going to be the same from manufacturing batch to manufacturing batch. So the regulatory agencies are really cautious about stability. They want stability and they want a surety that you can have consistency with your product from batch to batch to batch. Because of these codified guidelines, the development process can be quite long. Here I've written 12 to 18 months. So the actual manufacturing process might only take 2 weeks in your big fermentation tank and a week and a half in your purification suite, so less than a month. However, to get to that point in time where you can do it in less than a month, it's a good 12 to 18 months of hard work coming up with a process which is robust and it's producing exactly what you want. As I said, the safety, particularly early on, the regulatory agencies want to make sure that it's safe. They also want to know, not knowing that it's just safe, that if it works the first time in your phase 1, you want the same product with the same quality in your phase 2, just so you don't have an issue between working in one phase and not the other. It's certainly something which partners are interested in. I would say that for people who are not entrenched in the biologics industry, sometimes the CMC, it's undervalued, underappreciated, but certainly anyone who knows a lot about manufacturing biologics and just biologics in general, the big pharma want to know about your CMC, they want to know about your manufacturing, they want to know that you have it under control. So this is just a comparative of small molecules and biologics, and it is very, very different. So I've just listed a couple of things here. So generally the small molecules, they're generally produced by chemical synthesis in tubes and such. However, biologics, they're made in living hosts and obviously there's variability in living hosts. These are generally, as it says, small molecules, they're small. Biologics can be very large. And this is just a depiction here. You've got aspirin, which is very small, it's less than 200 in molecular weight. Antibodies over 150,000. So there's huge differences in the size of these molecules. Generally, the small molecules, they're generally singular entities. However, biologics are never ever single entities. It's a population of proteins which are very, very similar. They sit within certain specifications, but there is some heterogeneity there, unlike the small molecules. So the characterisation requirements for biologics are a lot more difficult and intense just because you know that you've got a mixture of molecules in your drug product. And that says it really here. There's lots of different modifications which can happen to a biologic. Not so many for small molecules, and they're generally known. Small molecules, once again, generally stable. Biologics are very sensitive to changes in condition. There can be changes in the amount of light, it can be changes in the temperature, it can be changes in the pH, it can even be shaking a biologic. So they're very sensitive, they have to be looked after well, and later on in the slide presentation I'll briefly talk about formulation, which looks at stabilising your biologic such that it's stable over 2-3 years of storage. Small molecules, this is once again all relative, easy to purify. Biologics, much more complex, because as I said earlier on in the presentation, there's a mixture of molecules there, and you're only interested in a certain range of molecules. So it's much more difficult and labour intensive to do the purification process. Low specificity with off-target effects versus high specificity. What I mean here and where this comes from, you've got a small molecule and it's binding its target. It's very small, so it can only bind to a certain area of the molecule. You've got a biologic, it's very large, so the surface area contacting between the biologic and your target is much bigger. So because there's much more area there, it's much more specific, requires more contact points. So generally, it binds only the target that you're interested in. That's not the case with small molecules, where you get a lot of promiscuity. They bind similar chemical structures. So this is really just going over what Robert said, so I can be very, very quick here. This is our molecule here. We've got the FC component, which is our half-life extension component, and that's fused to our eye body. So this is just a general outline of the process that happens in biologic manufacturing. I would say here it's drawn up in a linear fashion. It sort of is. While some of these activities are happening here, you're starting some of these activities, so it looks like it's very simple and it's very linear. It's actually quite a complex process. There are lots of wheels and cogs which all have to meet at the same time. So even though I'm going to depict it like this, it's actually much more complex than what this schematic is showing. So the first thing is cell line development. So this is really going to be your starting product. This is where you make your cell, which has got your DNA in it, which is going to tell the cell to make AD-214. You need a great cell line to start with. If you start with something poor, you're going to end up with a poor quality. And for this whole thing, I was trying to think of an analogy, and I'm going to go to wine because I do quite like my wine. Here, to me, this is like you've got to start with a good grape. If you start with a bad grape, you're not going to end up with good wine. And I know that as of yesterday because I tasted our CSO Mick Foley's home brew yesterday, and I can assure you, if you don't have good grapes, you don't end up with good wine. It was okay, Mick. So once you've got your cell line here, you go to cell line expression. So that is, how do we get the cell to churn out as much protein as we can at the quality that we want it to be at? And the way that's done is by monitoring certain growth conditions. You want certain amounts of oxygen, you want a certain temperature, you want certain pressures in the tank, you want the right nutrients, and all these things have to be optimised to get the cell to spew out all the protein at the right quality. So for me, going back to the wine analogy, that's like the grapes. You want to grow them in good soil. You want the right temperature. You want to pick them at the right time. So if you do all that, you have a chance of coming out with a good product. Once you've got your cell, it's releasing all your protein or your therapeutic into the media. But it's mixed with a whole bunch of other things. It's mixed with other cellular proteins. And some of the cells break open and release their own proteins. There's chemicals in the media. So there's a mixture of things amongst which is your protein. You've got to come up with a way to get your protein out and isolated from everything else. So once again, to the wine analogy, I'm walking to my bottle shop tonight. There's a whole bunch of different wines. How am I going to pick what wine I'm going to have? Well, I know I'm having a steak that night, so I'm going to filter out all the white wine. I'm only looking at the red wine. I know I'm going to be eating with John Westwick on our board. I know that if I hand him a Pinot Noir, he's going to throw it back at me. It's got to be a Shiraz. So I can filter out all these other wines, and I know I don't want the white, I don't want the Pinots, I don't want the Cab Sav, I want the Shiraz. So it's a little like here. I know what I want. It's in the tank there somewhere. I just have to come up with a process to get that out. And finally, I've got that protein. But I know that from a commercial perspective, it's got to be stable for generally at least 2 years. So you've got to put it in an environment where it's going to be stable for 2 years. And as I said early on, proteins are very sensitive. So you've got to test a whole bunch of additives to come up with something which is going to make it, stable. Back to the wine analogy. If I've got a lovely bottle of wine, bottle of red, I'm not going to put it out in my garage in the middle of winter. I want it in a nice cabinet at 18 or 23 degrees or whatever it's meant to be at. But I want it at a temperature where it's going to be stable. It's a bit like the formulation. I want it in a solution where it's going to be stable. So hopefully those analogies weren't too far off the mark. Looking here at the process, the cell line development, we've contracted that out to a company called Selexis, huge experience. All this component, the cell line development, the purification, the formulation, KBI Biopharma are the organisation which has been doing that for us or with us. So why do we pick Selexis? Why do we pick KBI? This was after quite an exhaustive process. We sent a tender out to many different CDMOs. We had detailed discussions with 12 separate CDMOs. Then we had extremely advanced discussions with 2 to 3 of our favoured CDMOs. We came up with Selexis for the cell line development. Why? Mainly because they've got great track record, 94 clinical drug candidates at all areas of the development pipeline, and importantly, they manufacture for 3 products which are on the market. It's extremely important. You want to go with a cell line that the regulatory agencies are familiar with, and you want to go with an organisation you know is going to provide you with a documentation package to support your application that the regulatory agencies are going to be happy with. So you go with someone who's done it many times successfully, and that's Selexis. It's the same with KBI. They've worked with lots of different kinds of products. What was particularly pertinent to us was the FC fusion. So there are lots and lots of monoclonal antibodies that lots of organisations have produced and manufactured. As Robert said, the FC fusions is not as prevalent, so we were really keen to go with an organisation who manufactured an FC fusion, and that was KBI. And on top of that, we've got these 2 separate organisations. We need to know that they're going to work together. They're owned by the same organisation, JSR Corporation, Japanese company, and most importantly for us, they together worked on over 20 cell lines. So Selexis have provided the cell lines, they've worked with KBI to come up with process development outcomes. So it's really important that they'd worked together before. This is an example of a few of the proteins that they've had out of the 20. You can look at the general titer. So this is grams of your product per litre of starting media. You can see they all sit around the 3.5 grams a litre. This is where our current AD-214 is at the moment. It's around about the 3 grams a litre. And what I'd like to point out here too is this is early on. As you go down the process, things get optimised more. And this is an example where at the start it was 4.5 grams, but by the end of all the optimisation, it was nearly double what it started with. So we're at the moment, this lead clone stage here, at around about 3 grams a litre. We're very happy with that. It certainly meets our expectations. So now I'm going to go into a little bit of technical discussions. And this is mainly around the cell line development, because we've pretty much completely finished that whole process at Selexis. So I'm going to outline what was done, why it was done, and explain what a clone is. So a cell line development, you have what's called a clonal cell line. So genetically, they're all the same. So how did Selexis go about this? Well, you've got to start with DNA. We've all watched crime shows, so we all know what DNA is, but just to extend a little bit more, this is a molecule which has the information to tell a cell how to make something. So what we have to do is we have to take some DNA, which has got the information in it, to tell the cell how to make AD-214. We have to introduce this DNA into a cell, which can then make the protein based on what the DNA is telling it to make. Generally in the biologics we use CHO cells, Chinese hamster ovary cells. So the first thing we do is we mix these two things together, the DNA and the cell. We then impose conditions which force the cell to uptake the DNA. So you end up with a mixture of cells, some of which have your AD-214 DNA in them, some which don't. We're not interested in these ones here, so we grow it under conditions where we get rid of the cells which don't have the DNA, and we end up with a pool of cells. Each of these cells now, they all have the AD-214 DNA in it. However, each of these cells are a little bit different depending on how the DNA was integrated, and they're all producing AD-214 at different levels and different qualities. So what we have to do, we have to be able to separate all these cells out and then look at them individually to be able to pick which of these cells in this big mixed population of cells is the one that's going to produce the most AD-214 at the quality level that we're interested in. So I'll show you in the next slide how Solexis actually go about it, but suffice to say just for now, we separate these cells all individually. We then grow them up in these little wells. At this particular point in each well, every cell is genetically identical. They're a clone. A clone is a copy. We then measure the amount of antibody which is produced. We pick our most highly expressing ones. We grow them up in a container which is a little bit bigger so we can see how these grow at a larger scale, and we can purify some protein and start to have a look at the quality. We go through that process a couple of times and at the end we end up with our lead clones. And as you'll see from the next slide, at the moment for Solexis we have 4 lead clones that all produce quite high levels. The final process is we need to check that these clones are stable. So we grow them for 60 generations to see that they're producing the same molecule at the end as they are at the start. And we have to show, this is a regulatory imperative, we have to show data to show that these are in fact all generated from the same cell line. And then we're ready to produce our therapeutic in these big tanks. So where are we at with Solexis? As I said, we're down to 4 cell lines. And this is where I'm just going to explain how they separate those cells from that mixed pool. So you take that mixed pool of cells, you dilute it, and then you plate it onto a plate in what's a semi-solid media, which is effectively like a bit of jelly that you eat at home. So it's a little thin tub of jelly, and you've got all these cells which are separated far enough apart that they're clearly distinct from each other. You can see that they've grown all from the same cell. So all these cells in this colony are identical, all these ones are identical. And what you do, you look for the amount of brightness or how much they glow, and the ones which glow the brightest are producing the most protein. So you can see the ones with the purple arrow, they're producing more protein than the ones with the orange arrow. And as I said in the previous slide, you then grow them into small wells, and then you expand it, and you come up with your final lead candidate clones. So this process has been completed at Solexis. It's gone extremely well. We've got our 4 lead clones. There's a number of things which have to be done for regulatory purposes. You need to confirm that the DNA sequence, which is encoded, is the same as what it was at the start. You need to say the copy number, determine the copy number, and you need to show that it's a monoclonal. That's really easily done from this image because you can see that these colonies are quite well separated, and the robot picks it out quite nice. That has all been completed and all went well. ICH compliant documentation, that's near completion. We're just waiting on the last stage of the cell line stability. So in a couple of weeks' time, this whole process is complete and we have our cell line. So it's gone really well. Moving to the next section, which is the cell line expression. So you've got your cell line, which we'll have in a couple of weeks. We want to identify the growth conditions such that we're going to get the most amount of protein per litre of media that we can get. So this is just a bit of an outline about how it's done. In this particular case, we're looking at product levels and quality. So we grow up our cells in these teeny little reactors. They're only little 15 mil reactors. You grow them all up and you look at the amount of protein that's getting produced by your lead clones. These are our actual 4 lead clones here. You can see these 2 here, which are just duplicates. They produce 2.7 grams a litre. These produce about 3.1, 3.2. And then what you do, you can take these, this one clone, and you can grow it up in different conditions. So all of these reactors here, it's the exact same clone grown up in different conditions. At the start we had 2.7. After we did the optimisation, you can see we're up to 3.3. So this is a clear indication how by coming up with the right conditions, you can increase the amount of protein that you're producing. Then we move on to the purification. As I said, the cell's producing a whole bunch of protein. It's mixed with a whole bunch of other proteins. You have to separate it. We do that by a technology called chromatography. I'll try and outline that here, hopefully in simple terms. We have these columns. They're just tubes. They can be glass tubes. They're filled with little beads, little porous beads, and each of those porous beads has a certain chemical group on it. So what you do, this black line here, you apply your mixture of proteins. As your proteins move down the column, each protein will have a different affinity for the resin. So some proteins will spend more time on the resin than others, and some will come off really quickly. And as it runs down the column, they all start to separate away from each other. And you can then, as they come out at the end of the column, you can start to collect them in a purified form. This gel is just an example of, this is the starting material, this is what it looked like. After one column, you can see that we've already filtered out all these proteins, we've filtered out that protein. So from one column, you've pretty much got rid of a good deal of your contaminants. Normally though, one column's not enough. You normally run it down 3 columns, each with little different chemical groups on them. You have these 3 steps. KBI has come up with a 3-step process which looks sensational actually. Finally, product formulation, as I said, sensitive to environmental changes. So we need to keep it in a solution which is stable and active over long periods of time. The early screens, we add additives, the technical term is excipients, but it doesn't matter what you call it, and you identify appropriate additives. We've done the first stage of that. It looks quite soluble. We're moving on now to the intermediate and the final steps of formulation. But certainly early formulation is looking promising. This is where we're at with the whole process. Our final cell line is going to be selected in the next couple of weeks. We expect the growth condition optimisations to be finished mid-March. Same with the purification and our formulation by May. And just ticking off the manufacturing milestones, I put a tick here. This is almost complete, but it's looking fantastic. It's going to go well. These are the future milestones, which is the optimisation of the development process mid-March. Tox material and the phase 1 clinical material is all on schedule as expected. Finally, I've said finally a couple of times, there is another slide. We've got manufacturing going to be finished Q2 2019, tox studies start Q3, and then the phase 1 Q1 2020. And during this time, Sam's going to be out there doing a lot of BD and getting us lots of partnerships. Manufacturing summary, it's got broad anti-fibrotic and anti-inflammatory effects, and it's safe. Our output has gone up from initially reported 1 gram a litre and now up to 3 grams a litre. We're on track to deliver our toxicology material. We're on track to deliver our phase 1 material. Our Delta is working successfully with KBI, and once again, my first sentence is my last sentence. Everything's going well. The cell line development's gone well. The purification's going well. That's it.
Great. Thanks, Dallas. Any questions for Dallas?
Dallas, just 3 grams a litre.
Yep.
What do you think you can get to, or is that good enough for a commercial product?
Commercial always depends on what your dosing is going to be, but 3 grams a litre is typical of lots of molecules on the market. If you look at probably 7 or 8 years ago, 1 gram a litre was exceptional. If you've got an antibody, it's not unheard of to be up at the 5, 6, 7 grams a litre, but for an FC fusion protein, this is pretty much sitting exactly what you'd expect. Yeah.
And just, you've outlined a couple of steps in that manufacturing process. Where is the risk that you see in each of those steps?
Robert mentioned early on that aggregation with an FC fusion protein can be a risk. Luckily, in the chromatography series that KBI has developed for us, there's a clear step in there which removes any aggregates which are present. So that being potentially a major issue, we know we can remove those aggregates. With anything, as far as the stability goes, you hope for the best, but they're long experiments. You've got to sit these things for 24 months, so you don't really know until the end of the 24 months. However, having said that, there's a lot of work which goes into selecting your lead formulations, and generally everything's fine. So at the moment, you can't say with 100% certainty, but I'm saying I've done a lot of these projects. We're sitting in an area where I'm quite comfortable at the moment.
Do you retain the IP on your manufacturing process, and should you want to or need to, could you transfer it to another manufacturer?
Yes, we can transfer it to whoever we want, as long as they have the appropriate facilities. KBI do not own any IP whatsoever. It's just a process. It's a fee for service.
Follow up, what kind of scale are you at the moment, or is it still too early in terms of size?
Yeah, so a lot of work's been done with 50 litre ferments. The tox material will most likely be done with a 200 litre tank, and then the GMP phase 1 material probably a 1,000 litre tank. What I would say though, generally most people are comfortable when you scale less than 10-fold. So 50 litres, you're certainly extremely comfortable going to the 200 litres and the 500 litres. What I would say too is the AMBER systems which we use, those little mini reactors that I showed you before, where there's 15 mils in them. Part of the development process is in larger ones at 250 mils. These tanks have been shown to be extremely indicative of what you see when you scale up, because they have really good temperature control, good pH control, good metabolite control. So these days it's a little bit easier because the technologies are there to give you good information early on that as you scale up, things should be fine.
Is this always going to be a batch procedure even at the commercial level, or is there a possibility of doing continuous culture? I mean, is the genetics stable enough to do that?
So continuous culture is something which the industry is looking at, but hardly anyone does that at the moment. The industry is completely driven at the moment by fed batch culture.